Good evening and welcome. My name's Luan Ismail. I'm the Executive Director of the Convergence Science Network, and a warm welcome to you to our presentation this evening. We're fortunate this evening to have a husband and wife team sharing their research with us. Sara-Jane and Mark Dawson completed their undergraduate medical training in Melbourne before travelling to Cambridge in the UK to undertake PhD studies. And returned to Melbourne in 2014. I'll pause there and invite them both to come up and perhaps tell you a bit about themselves. So please invite Sara-Jane and Mark Dawson.
Thank you. Can you hear us okay?
Great. So I'm Mark Dawson. I, as Luan just explained, am a clinician scientist at the Peter Mac. And I guess today is in part to tell you about how we got into what we do today and what that journey was like, and then to explain a little bit more about exactly what we do at the Peter MacCallum Cancer Centre. So I'll start and then I'll let Sarah have a go. So I did my undergraduate at the University of Melbourne, and it was very clear early on for me that I was wanting a little bit more than just clinical practice. So between my 5th and 6th year of medical school, I decided to take a break at that stage and really go and find out what life as a scientist would be like. And in part that involved a time at the University of Oxford and also some time at the Ludwig Institute and the Walter Eliza Hall. And I'd say that that was, a year that really changed my perspective on what my future would bring. I worked primarily in a model organism, which is called zebrafish, and I studied how cells go through the cell cycle and how they replicate themselves. And it really brought to me a thirst for knowledge in scientific discovery that has stuck with me all the way through. I would say I was a very reluctant clinical practitioner, and had I never met Sarah, I probably would never have finished medicine. But having made the decision to complete medicine, I'd say now that I can always see myself as a clinician. I enjoy treating patients, speaking to patients, and it's a part of my career that I would never let go, and would have been a mistake early on to have forfeited this. And hopefully I can tell you during my talk as to why medicine is just as important to science as science is to medicine. So I'm going to stop there and hand over to my good wife.
So my journey was similar but somewhat a little different to Mark's. I also was from Melbourne. I did my medical training in Melbourne and continued to specialise and decided that I was very interested in cancer. I enjoyed looking after cancer patients and really completed my medical and specialty training in medical oncology. But I guess towards the end of that specialty training, I started to develop more of a thirst for science, thinking that, through discovery, we may be able to make better advances for our cancer patients. And it was at that time that Mark and I decided to move to Cambridge. We'd been well trained in medicine, but felt like we needed to train ourselves as scientists. And we're very fortunate to have the opportunity of going to the University of Cambridge, where we both did PhDs and then stayed on to do postdoctoral research. We were there for 7 years before relocating back to Melbourne and to the Peter MacCallum Cancer Centre, where we now work. So it's wonderful to have the opportunity to talk to you today about the research that I feel passionate about. As a medical oncologist, I see patients. I primarily specialise in breast cancer management, so I look after patients with breast cancer. But on a research front, I'm very interested in developing new tools that we can apply in the clinic to help with the decisions that we make for cancer patients every day. Decisions around what might be the best therapies for patients, understanding whether patients are responding to treatments and when to change those therapies. And the area I guess I've become very interested in is what we can learn from blood tests and how we can apply this knowledge in the clinic to learn something about cancer behaviour. And we've actually known for a long time that the blood can tell us a lot about cancer. I think it's quite remarkable to reflect upon the fact that it was in the late 1800s that we first discovered that you can identify cells in the bloodstream that resemble cancer cells in patients that have cancer. So this is the concept of the fact that cancers The cells themselves can enter the bloodstream, and it's through this process that cancers can unfortunately spread to other parts of the body. And that finding was actually discovered by a Melbourne physician, Thomas Ashworth was his name. He's in this picture at the back here, and this photo was actually taken at the University of Melbourne. But in addition to the concept of circulating tumour cells that we can find in the bloodstream, we've also identified that it's possible to find what we call cell-free DNA. This concept was discovered in the late 1940s, and it was soon recognised that cancer patients have much higher levels of this cell-free DNA in the bloodstream because the tumour itself can release part of its DNA into the blood. And the idea of potentially using this in cancer patients to follow some information about that cancer has really only started to take hold in the last 10 years as we've had better technologies to be able to measure and quantitate this really small amount of circulating tumour DNA that we can find in the blood. And part of the reason why this field has advanced so much in cancer actually stems back to some pivotal work that has also been done in the field of prenatal genomic diagnostics. You may be aware that it's now possible for a pregnant woman to have a blood test done. Fetal DNA can actually be assessed in that blood test, and that can be used to identify particular genomic disorders. Trisomy 21 or Down syndrome is the common test that's now done through assessing this fetal DNA within the maternal circulation. And it was when these studies were first published, sort of around 2010 to 2012, that we started to get an idea that actually a very similar concept may apply in cancer. If we could take a blood sample from a patient with cancer and look at this cell-free DNA in the circulation, we may be able to learn something about the tumour from that information. So how does this cell-free DNA actually enter into the blood? Well, it's really thought primarily to be through a process of apoptosis. And that's really where cancer cells are turning over, they're growing rapidly, they're dying in the patient, and they're releasing their DNA into the circulation. And we can then study that DNA in great detail to try and understand something about the characteristics of that tumour DNA in that patient. The characteristics of the DNA that we see in the bloodstream are very specific. We see really short fragments of this DNA, and that certainly has some implications for how we go about to do this kind of analysis. But why has there been so much excitement in this sort of field of research in cancer? Well, it's really this concept that these tests may be able to provide us a liquid biopsy alternative to tissue biopsies. So tissue biopsies are something that are frequently used in cancer management, and that really involves usually inserting a needle into the site of the cancer in a particular individual and taking a sample of tissue. And that's very important because it's helpful to make the diagnosis of cancer, and we learn a lot about the cancer by being able to study those cancer cells down the microscope. But unfortunately, tissue biopsies are not without limitations. They're invasive. Of course, these procedures are very difficult for our patients to undergo. They often can't be performed regularly through someone's illness, and we know that cancers are not static. Cancers change over time, particularly when patients are exposed to different therapies, and we need better ways of being able to assess how that cancer is changing or evolving over time. And certainly not practical from an invasive tissue biopsy perspective to do that frequently. It's not uncommon that unfortunately we may be looking after patients that have advanced cancer, like this picture on the right-hand side where a patient may have cancer affecting multiple different sites within their body. In that sort of circumstance, it's also impractical for us to biopsy all of these different sites. And we also know that cancer can be very heterogeneous. The cancer may not be the same at every different location. So if you're relying on a single biopsy, then that may not be representative of the whole cancer. So can we do better with a blood test? If we can follow this tumour DNA in the blood that's being shed from all of the different sites of the cancer, then that may give us a more comprehensive or global perspective on the changes within the tumour. And then we have the advantage of being able to follow this from a simple blood test that's minimally invasive. Most of our patients are having these blood tests regularly through their management, so it can be really repeated very frequently to give us great temporal resolution about these changes over time. But unfortunately, it's not without its challenges. Trying to identify this tumour DNA in the bloodstream is like looking for a needle in the haystack. We all have this cell-free DNA in our bloodstream. Most of it's coming from healthy cells in our body. But in a patient with cancer, only a very small fraction of that cell-free DNA is being derived from the tumour. So what we really need to tease out is that tumour DNA compared to all of the normal DNA that's in the bloodstream that's being released from our healthy tissues. And I think that's really the reason why the field has progressed very, very rapidly over the last 5 to 10 years, because our technologies have improved. We now have much more sensitive genomic technologies to really be able to drill down and measure these really tiny quantities of cell-free DNA that we find in the bloodstream. The different kinds of analysis methods that we use are complicated. There's no gold standard. Many of them involve standard laboratory techniques like PCR or next generation sequencing tests that are now being commonly applied in cancer to be really able to analyse genomic changes within a cancer. The choices between these methods really depends on what we're trying to understand when we do this kind of analysis. But there's two critical pieces of information that we obtain once we do this kind of testing on the blood. The first is we have a quantitative analysis. We obtain some understanding of the amount of circulating tumour DNA that's in the bloodstream, and that can tell us something about the tumour burden or the extent of cancer in an individual. But on the other hand, on the right-hand side, we also get this amazing ability to characterise all of the genomic changes through this blood test that can tell us something about the genomic features of the cancer. So how can we think about applying this in cancer management? I think this is the other very exciting aspect of this technology. We can actually think about clinical applications in all facets of cancer management. That could really come from the beginning. Could we think about using these tests for early diagnosis? Could these tests be applied as screening approaches? Once patients are diagnosed with cancer, can it tell us something about an individual's prognosis? If you know something about the levels of this circulating tumour DNA, will that tell us something about the likely prognosis of this individual from their cancer? Once patients are started on therapy, can changes in the levels of this circulating tumour DNA tell us about how patients are responding to different therapies? When patients have completed their therapy, many patients are undergoing surveillance because a proportion of patients may relapse over time and their cancer may come back. Could we use these tests to detect if there is any minimal residual disease left behind and identify which patients may be at highest risk of relapse? And lastly, this concept of molecular profiling. Can we really use this technique to get at all of the different characteristics of an individual's cancer so we can hopefully prioritise therapies more effectively? The other exciting aspect of this technology is that it's not only relevant to one single cancer type. The majority of cancer types actually shed their DNA into the circulation, so it's a test or a technique that may be relevant across many different types of malignancies, ranging from majority of solid malignancies, breast cancer, colorectal cancer, prostate cancer, lung cancer, to name a few, and also hematological malignancies. This slide just looks at a range of different cancer types and the ability to find circulating tumour DNA in patients who have advanced disease. And it's certainly readily identifiable in the majority of patients with cancer. So I wanted to share, I guess, two different clinical scenarios with you briefly today, just to start to explain to you about some of the applications where we're currently applying this in cancer management. And the first sort of stories that I wanted to share with you are in breast cancer. Breast cancer is certainly the patients I treat in the clinic and where I started to think about applying this kind of technology. This work that we first published was the first body of research that I did in this area. This paper was published now back in 2013 when I was still in Cambridge. And we were looking at patients who had advanced breast cancer, and we were following levels of circulating tumour DNA in these patients to try and understand what this could tell us about how patients were responding to therapy. And we learned some really amazing findings from this study. It was the first time we tried to apply this in breast cancer patients. This figure on the left is a figure of a particular patient. She was undergoing therapy for breast cancer. The different therapies are shown by the shading there. And the circulating tumour DNA is the one that's in blue. And it's just following the levels of this, whether that's rising or falling through these different therapies. And you can see that the blue line falls very rapidly when the patient was exposed to a particular type of chemotherapy. The patient responded well to that treatment. The circulating tumour DNA was falling. But then when she was switched to a different therapy, you can see that the levels started to rise again, and she was unfortunately progressing on that treatment. So there was this relationship between the levels of circulating tumour DNA and understanding how this response was happening. The second most exciting finding was that the changes in circulating tumour DNA levels often happened before we saw evidence of disease progression on an imaging scan. So many of these patients usually have follow-up with serial radiological scans. They often have CAT scans done on a regular basis to try and understand whether they're responding to therapy or not. And this example on the right shows that the circulating tumour DNA was starting to rise before we saw evidence of the fact that the patient was progressing on that therapy. So we had this window where we could see that the circulating tumour DNA was rising, and the window was about 5 months on this study, where we started to get an inclination that the patient was no longer responding to their therapy before the evidence was established on radiological imaging. And 5 months is not an insignificant amount of time for someone to be remaining on an ineffective therapy. If we know that information earlier, then we can do something about it. We can potentially change them to a different therapy that may be more effective. So that was certainly another very exciting finding that we developed from this research. And then we've gone on to characterise this kind of work in a lot more detail. We've certainly done much more complex studies now where we've really tried to understand just how good are these blood tests at providing a liquid biopsy alternative to tissue biopsies. This was one particular complex study where one of the patients that I was looking after in Cambridge very generously consented to an autopsy being done after her death, where she agreed to multiple areas of her cancer being sampled, so we could study those in great detail, and we could do very complex genomic analysis on all of those different areas of the cancer and really understand the features of those and whether we could detect those in the bloodstream. And it was really remarkable, the overlap that we could see. Many of the different changes that we could see across these different sites of the cancer were well represented in blood, giving us the confidence that this really may be able to provide a liquid biopsy alternative to a tissue biopsy in some contexts. And that's now been followed up by many studies internationally. There's now been thousands of patients that have had these kind of tests done, really comparing just how good a tissue biopsy and a liquid biopsy correlate. This particular study was of many patients, quite a large number of them had breast cancer, and the concordance with tissue biopsies is as high as 90% and can be even higher when your blood sample and your tissue sample are collected at exactly the same time. So the concordance is great. So if we can think about replacing some of our tissue tests with this kind of analysis, then that would certainly be a very advantageous thing for us to do. We are now in the process of trying to get a better understanding of whether targeting particular genetic changes that we see in the bloodstream with particular therapies will be as effective as if we were targeting mutations that we identified from tumour tissue. And that's certainly something that's being worked on now in the context of clinical trials to try and answer that question. Here locally at the Peter MacCallum Cancer Centre, we are doing this kind of testing on many patients that come through the clinic with advanced breast cancer. Many of these patients may have exhausted all of the standard therapeutic options that they have available and looking for alternative treatments and trying to make decisions on whether they should be involved in particular clinical trials. In these patients, where possible, we try and obtain a tissue biopsy, but it's not always feasible, and we're always able to collect a blood test. So we take that blood test and we do some circulating tumour DNA profiling for particular mutations. In this example, the names of the mutations are not so important, but the group of them is listed on the right-hand side. But through this kind of work, we can actually identify many mutations just simply from this blood test. And as a result of that, we can help guide and suggest to patients which clinical trials may be most appropriate. So one example in this scenario, one of the common mutations that we're identifying is in a gene called PIK3CA. And we have a clinical trial at the moment of a targeted therapy, a PI3 kinase inhibitor, and which we understand is certainly more effective in patients that carry this particular genetic change. So if we can identify that genetic change in the bloodstream, then we can prioritise patients for this particular clinical trial. And certainly that kind of approach has been very successful, and we've been doing that routinely in patients with advanced breast cancer. The other area in breast cancer or other cancer types that I think will be very exciting is this concept of surveillance or tracking minimal residual disease. I alluded to it earlier, where patients come to the end of their treatment, which we hoped to be curative, but unfortunately sometimes patients will relapse after they have undergone that curative treatment. And at the moment we don't have good ways of often understanding which patients may be at highest risk of relapse. Certainly in breast cancer we don't have good tests to do that, and we don't have good tests to follow patients when they've finished their treatment to know whether they're at risk of their cancer coming back. These particular molecular tests may be helpful in that respect. Could following the circulating tumour DNA tell us something about which patients may have some minimal residual disease left behind, maybe at high risk of relapse? And if we could identify that, then hopefully we could do something about it. We may be able to prioritise more intensive therapy for that group of individuals. There's certainly some exciting studies that have been done internationally on small groups of patients that have suggested that this is possible. This particular study showed that if you had all of your treatment for breast cancer and you came to the end of that and you had a blood test, and if that blood test was positive for the presence of this circulating tumour DNA, then you had a much higher risk of your disease coming back. So we're now running similar studies at Peter Mac to try and identify whether in a larger group of patients, whether we can do a similar kind of analysis and really identify which patients may be at risk. And I guess this is what we're hoping to achieve. At the moment, we don't really have good ways of picking out the purple patients on that left side. The purple patients are the ones that are at highest risk of their disease coming back. But potentially we could identify those kind of patients with a molecular test, such as the blood test that I've explained. And in doing so, we'd hope to achieve two aims. We might be able to de-escalate therapy for patients who don't need it to try and reduce toxicity and costs. But we could also potentially prioritise more intensive and novel treatments for those most likely to benefit. So in the last few minutes, I just wanted to change tack completely and show you some applications in a very different cancer type. I've given you an example of breast cancer, and that's just one solid cancer where this kind of technology may be helpful. But we're also starting to understand that these kind of tests may be useful in patients with hematological malignancies. This particular example is in patients with myelodysplastic syndromes. Myelodysplasia is really a cancer type that affects the bone marrow and stops the bone marrow being able to produce normal, healthy blood cells. To diagnose this condition, unfortunately, we rely on bone marrow biopsy, so that's a very complicated and difficult procedure for patients to undergo. And one of the things we've learnt from all of the study that we've done of cell-free DNA is that we know that in most of us, the cell-free DNA that we have in our bloodstream is actually being derived from our bone marrow. It's being derived from hematopoietic cells. So we wondered before we started this study whether in this particular cancer type, in myelodysplasia, whether we could actually identify really high levels of the tumour DNA that was being released from the bone marrow into the blood. Like with many cancer types now, we actually have a much better understanding of all of the genomic characteristics of the cancer. The absolute names and particular genetic changes on this slide are not important, but this is a common feature now through sequencing studies that we've been able to get much better resolution and understanding of all of the genomic changes that may be occurring in a cancer. And we can now take this information that we've learnt from biopsies and actually apply that to the blood and see if we can identify these kind of genetic changes through our simple blood test. And in this particular study, we did that in a group of patients who had myelodysplasia that were being treated with particular therapies. And what we found was really remarkable. We could really find very high levels of these particular genetic changes or mutations in the blood. And the correlation, if we did that kind of testing on a bone marrow biopsy versus a blood test, was highly concordant. You can see that the concordance was about 84% on this occasion. So the information that we were obtaining from the bone marrow biopsy was almost identical to the information that we could obtain from the blood test. And I'm sure if you asked most of the patients that had gone through these procedures, they'd much rather have a blood test than a bone marrow biopsy to obtain this information. We can follow particular mutations that we know are important in this disease. This is a particular example of mutation that we were tracking in a particular patient. This was a patient who had had very difficult bone marrow biopsies along the way. They'd had bone marrow aspirates, but they were suboptimal. We hadn't really got a lot of information from them, and that's commonly the case. Not all biopsies that are done give you the effective information that you need. But in this particular case, the blood tests were much more helpful in being able to direct that information. And lastly, as well as being able to identify specific genetic mutations through the bloodstream, we can also start to look at broader genetic changes, broader chromosomal changes that can be identified in this patient. This is just a particular example of a patient that was in this study showing the particular chromosome patterns in this patient. And this particular patient, you wouldn't have picked this probably from the back of the room, but they had an abnormality on one of their chromosome 9. There was a change there that had been identified from their tumour tissue. And what's exciting about our blood testing technology is that we can start to identify some of these changes in the bloodstream. This plot just looks at all of the different chromosomes on this patient, and these were 3 different blood tests that were taken. At baseline, when the patient was diagnosed, they underwent treatment and went into remission, and then their disease came back again. I don't know if it projects very well, but you can see that little yellow change on chromosome 9. That was a deletion that we were able to identify on chromosome 9, and we could pick all of this up from the blood test rather than relying on the bone marrow biopsy. It was there at the beginning, it went away when the patient responded to therapy, and then it came back at the time that the disease came back. And we can couple this with this mutational analysis that we also do in the background to obtain an enormous amount of genomic information about the cancer. So just lastly, I guess, where is this kind of technology in terms of applying it in the clinic? Well, I've shown you some examples where we're already starting to do that, and I think we're really close to starting to see more of these tests be integrated into routine clinical practice. I think the area of need greatly at the moment is demonstrating what we call clinical utility, so proving that if we make treatment decisions on the basis of these tests, how likely is that to improve patient outcomes? So if you're changing therapy on the basis of one of these tests, is that going to lead to improved survival for patients with cancer? And to really answer those questions, we need large clinical trials that are really going to take national and international efforts to try and do that. But I think the technology is ripe now, and I think we will see these tests integrated into cancer management in the future. So I just wanted to thank everyone involved in this research. Certainly run a very big lab now. There are many individuals that make this kind of research possible. It's very much a team effort. So it's a huge thank you to all of the team that we've built since we started up in Melbourne in 2014. I'll just introduce Mark now to take over. So.
She's always been a tough act to follow, but I'll do my best. I also work at the Peter MacCallum Cancer Centre, as I alluded to earlier. My work is very different to Sara-Jane's. We work in a field called epigenetics, and hopefully I'll aim in the next 20 minutes or so to try and give you an overview of what this field is, why we're so interested in it, and why we think it holds the key to potentially changing the natural history of some of the most aggressive cancers we treat. So as I said to you earlier on, I am a haematologist. I'm a blood doctor and see primarily patients with blood cancers. As a consequence of the career I've chosen, which is to couple medicine with science, I've had to really focus my area of clinical activity. Now, primarily the only patients I see are patients who suffer from this disease called acute leukemia. Acute leukemia has two major different types. There's acute lymphoblastic leukemia, which is primarily seen in children, but the adult disease, acute leukemia, is primarily a disease called acute myeloid leukemia. And so each year about 90,000 patients worldwide are diagnosed with this disease. It's actually genetically a very simple disease in contrast to the mutations that you saw that Sarah showed you, where we have, diseases like melanoma and lung cancer that have many hundreds, if not thousands of mutations. Acute myeloid leukemia can be driven with as little as one chromosomal translocation. But what really happens with this disease is that it leads to a block in differentiation and an increase in the ability of the cells to self-renew themselves. Now, when patients come to see me when they are diagnosed with this disease or potentially getting diagnosed with this disease, it is in many ways a medical emergency. We don't have a lot of time to procrastinate about what the best treatment options are. We have to get on with treatment relatively quickly. Untreated, the life expectancy with this disease is measured in weeks to months. The problem with getting on with treatment very quickly is that we haven't had great treatments for over 40 years. So it's been 40 years since we've had a new drug approved for the treatment of this disease. And that's not because we do very well. In fact, what's shown up here are the survival curves for patients all over the world diagnosed with this disease. If you had a perfect treatment, you'd see a straight line that goes across here where we cure everyone. And to summarize this, what these survival curves tell you is that we cure about 1 in 4 to 1 in 5 patients with this disease. Okay? It is a very expensive treatment because it is one of the most aggressive chemotherapy cocktails we can give another human being. The patients are in hospital for 30 days waiting to recover from the chemotherapy, and it is very expensive for something that is not very good. Okay? Now, I said we cure 1 in 4 to 1 in 5, but we actually get about 80% of patients into what we call a complete remission. What does that mean? And Sarah talked to you a little bit about that. It means we don't, through the best available methods we currently have any evidence that there is any residual leukemia left. But we know that even though we get 80% of patients into remission, that only this is sustained in 20%, 20 to 30%. Okay? What happens when the leukemia comes back? Well, the prognosis is even more grim, okay? In this disease, if you relapse with leukemia and you're older than the age of 55 to 60, it is essentially an incurable disease. Why 55 to 60? Because the younger patients are able to tolerate the intensity of the major salvage at this stage, which is that of an allogeneic bone marrow transplant. Okay. So what the major problem with this disease has been that we have treated it as a single entity. We think that everyone with acute myeloid leukemia should respond to the same chemotherapies. And now in the era that we live in, we understand that this is far from the truth. In fact, the cancer in the same patient can be very, very different. Okay? So let me give you an overview of normal blood cell development and give you some simplistic ideas as to how this goes wrong. Okay? So every tissue in our body that you can think of has a hierarchical structure to it. And that's no different for blood. And at the apex of this hierarchy are a rare population of cells that we call stem cells, and they're tissue-specific stem cells. In this case, they're blood stem cells. And these stem cells have two unique properties. They have the ability to replicate themselves and become another stem cell, and then they have the ability to then give rise in a very organized way to all of the other blood cells that you and I know about. Red blood cells, white blood cells, platelets, etc. Okay? What we think goes wrong in leukemia is that a mutation arises either in the blood stem cell that co-opts the ability of the cell to renew itself, or it reinstates it to one of these progenitor cells, but then it blocks the ability of the cell to make a decision to differentiate, to become a mature blood cell. And then you get this increased self-renewal and your cancer gets bigger and bigger. Leukemias also have a hierarchical structure, and at their apex are cancer stem cells or leukemia stem cells. It's the red cells here. Right? And it is these rare cells that sustain the leukemia. So why do we get 80% of patients into remission? Because chemotherapy is very good at pruning the tree. Okay? And so we get rid of the bulk leukemic population very effectively. But in the majority of patients, we don't eradicate these leukemia stem cells. And this then sits there, it adapts, and it comes back when your cancer comes back. Okay. Why is this knowledge important, and why do I care about epigenetics so much? Well, let's start by what is epigenetics. Okay? And by and large, I know it's a mixed audience with some very astute scientists and some very general people in the audience as well. So I often explain epigenetics by giving you one of the best recognizable examples of epigenetics. Sarah didn't talk about our children, but both our children know that the caterpillar becomes the butterfly. But what they probably don't know is that the caterpillar and the butterfly have an identical genetic code. There is not a single base pair that's different. Okay? But they look very, very different. So it is how that genetic code, those genes, are accessed at the right place at the right time that gives you these two very different phenotypes. You know, to a layperson, this might seem like two different organisms. Okay? And it is studying the way our DNA is accessed at the right place at the right time to give organized gene expression, to repair your DNA template, and to replicate DNA. That is the study of epigenetics. If we now move to the world that I spend most of my time thinking about, and this is blood cell development. So this is, if you open up any medical textbook, you'll see this. Right? And there is a problem with this slide. And the problem isn't that we have missed some rare cell that has just been described last week that is not quite on this list. The problem lies in the arrows. The arrows are all pointing in a unidirectional way. They suggest that cell fate determination, when this cell makes a decision to become this cell, which then makes a decision to become this cell, it is a one-way street, right? And we know that that is just simply not true. Okay? And we've known for over 60 years that this is simply not true. We know that normal development, by and large, goes down this pathway. You know, it is easier to make a decision to go down one pathway and it is easier for a cell to commit with the right cellular context to become a different type of cell. But we can reverse this, right? And we can take cells back. We can make one blood cell become a completely different blood cell, so-called transdifferentiation. And the discovery that cell fate is not a one-way street led to the Nobel Prize for these two individuals. Shinya Yamanaka, who showed that he can forcibly express certain genes to drive cells backwards, and John Gurdon, who showed this in the late 1950s and early 1960s. Who in the audience has heard of Dolly the sheep? Just show of hands. Now keep your hand up if you'd heard of John Gurdon. Okay, right. So Dolly the sheep is John Gurdon's experiment just done on a sheep. Okay, and it is probably the best epigenetic experiment that will ever be done. Okay. John was, I worked at the institute where he was at, and he was actually my PhD examiner. So he examined my PhD after it was done. And I remember actually going to the pub with him after that, and I asked him why he did this experiment. And so here's the experiment, okay? And he said, Mark, at the time, the prevailing opinion in the field was that when a cell made a decision, when a cell made a decision to become a skin cell, it lost the DNA, it needed to become a blood cell or a liver cell. It didn't need it anymore. It had made a decision. And I thought that this was a very ineffectual way for evolution to have worked, right? Why would we just get rid of the DNA? And that's a very complicated process to try and explain. It was more likely that that DNA was always there, it was just inaccessible. So the only way I could think of doing that experiment was to do this. So what he did was he took a mature cell, an intestinal cell, and he took out the nucleus from this intestinal cell, and then he took an oocyte. He destroyed the nucleus of the oocyte and put this nucleus in the right place at the right time, give it the right context to see what happens. And what happened was a tadpole happened. And this unequivocally says that you didn't lose any of this DNA. It was all there. It was just shut off, and it needed the right context for it to be re-expressed again, for organized development to happen. And this, by and large, is exactly what epigenetics is. Okay, so let's drill down a little bit more. So this is the nucleus of our cells. We have many trillions of cells, and I said to, if any of you read the abstract, I gave you a context of what we're dealing with. Right? So if we were to take out the DNA from any one of you in the audience and stretch it out end to end, you have enough DNA to walk to the sun and back 600 times over. Right? So imagine the problem. How do you package this? How do you replicate this? How do you repair this? How do you do organized development in different tissues? Right? And so that is the study of epigenetics. Right? And there are some basic concepts that help focus us. By and large, the areas in a cell that need expression to tell this cell that it needs to be a skin cell, well, those areas of the genome are much more open and accessible, so-called euchromatin. And this isn't just a theory. Up above that is the electron micrograph. And the Catholics in the audience will see this as rosary beads, right? And that's what they look like, right? And, the areas of your genome that do not need to be expressed, that can be quite tightly compacted, well, that's called heterochromatin. And so, in your skin cell, these are the genes that are required to become a blood cell. You don't need it, but it's there, it's not deleted, it's just shut off. And the basic fundamental unit of chromatin is DNA wrapped around a bunch of proteins called histones. Well, this just repeats over and over and over again. So if we now drill down to this, what we now see is that these proteins, these histones, as well as your DNA, are chemically modified. And these chemical modifications, much like dressing on your Christmas tree, they're not there just for decorative purposes. They actually serve a very instructive role. They help organize the way our DNA is accessed. Okay? And the key to this is if you go to yeast that you help brew your beer, and you look at the proteins there around which they have histones as well as around which DNA is wrapped, the histones there are modified in exactly the same way on exactly the same amino acids in yeast as they are in humans. What this tells you is that this is incredibly conserved and incredibly important. So how does this work? So here's your histones, and what happens is we have a group of proteins that come and lay down these chemical modifications called epigenetic writers, and they put this down. Epigenetics, unlike genetics, is very dynamic. So there's enzymes that come and remove these modifications called epigenetic erasers, because once your gene's expressed and you need to turn it off, bang, it's gone. How does these modifications work? We have evolved proteins that have specialized domains that come and bind and recognize these modifications. These are called so-called epigenetic readers. And they serve the purpose of bringing in the big machinery to the right place at the right time to move around your histones and nucleosomes to access your genes for DNA replication, DNA repair, and gene expression. Now, I know that you've had a number of eminent scientists speak in this lecture series, and Convergence Science has been great at doing that. And you may have seen this slide. It's from the highest cited paper that has ever been published. It's by Doug Hanahan and Robert Weinberg. It's called Hallmarks of Cancer. And it tells you about all of the different things that can go wrong to give rise to a cancer. There's been 2 iterations of this article, and if you read it end to end, you will never find the word epigenetics. You'll never find the word histones, chromatin, or anything else like that. And originally when I was a student and I read this, I was really quite upset that they'd missed the point. But then I realized that actually they couldn't talk about it because epigenetics was important for all of these. Right? Because every single one of these hallmarks requires access to your DNA to be able to change the cell making a specific decision. Right? Be it causing the cell to die, increase its blood flow, etc., etc. Okay? All of this involves the chromatin template. What we've learned by sequencing cancer genomes over the years is that the most common class of genes mutated in cancer are those that involve laying down, removing, and reading these modifications on the chromatin template, so-called epigenetic writers, readers, and erasers. So that's a discovery that has been made in the last 10 years, and it's come from sequencing efforts from a number of people around the world. And it has helped focus the pharmaceutical industry, academia, and clinicians in trying to leverage this because, as I said to you, the epigenome is very dynamic. So maybe we can change it to make cells make decisions we want them to make. And that, by and large, is what we try to do. Okay? And here, listed here, are all the new drugs we've developed in the last 5 years to do precisely this. Okay? And everything here in red is a drug that has now gone through the preclinical evaluation and is now in clinical trials or actually being used regularly in the clinic. And we've played some role in this process. Okay? So when I was still in Cambridge, we developed a drug that was the first in its class to do this. So it helps block a protein recognizing one of these chemical modifications, a so-called epigenetic reader, BRD4, and in doing so, stops the genes that have been abnormally mutated to drive the cancer from getting to the genes to turn them on in an aberrant way. Okay? So this example of blocking a protein-protein interaction and turning genes off was one of the first examples of how we could do this. This was published in 2011. It was a very competitive time. There were a number of other groups working around the world to show the same thing, and in fact, 3 other groups showed exactly the same thing within a couple of months of each other. And this was really important because it really helped establish the validation and clinical platform for taking this into the clinical arena. And one of the great examples and merits of being a clinician scientist, one of the great joys of my job and Sarah's job is that not only are we able to make the discoveries in the laboratory, we're able to see this now translate into the clinical arena. And when we moved to Melbourne, I brought one of these clinical trials with us here to the Peter Mac. And it is this trial here which is one of the largest clinical trials with this drug in advanced blood cancers. Now, some of that data has started to come out, okay? And this is what's known as a waterfall plot. And let me just explain what this means. If your cancer did very well, you got a line that goes underneath the axis here, the x-axis here. Let me just try and get this up here. So these patients, for instance, they had a complete remission to the drug. Whereas these patients, they derived very little benefit on the other side. Okay? So if you look at this waterfall plot, you say, well, this is a really mixed picture, right? And by and large, this is kind of what we expected. So this is not a drug that is an earth-shattering drug, but it does have responses, remarkable responses in some patients who've failed everything else. Okay? And that, I guess, leads me to my next slide, is what my day job is. Okay? So as a clinician scientist, I'm really interested in being on this side of the spectrum, right? Trying to find new therapies that we take into the clinic. We understand what's good about them and what's bad about them, and then we go back to the lab to try and make them better. And how do we do that? Well, we use a range of cutting-edge technologies. I know Jennifer Dudana spoke in this lecture series a while ago, and she talked about CRISPR and gene editing. We use that every day, right, to try and find new targets. And then we work with the commercial sector in various aspects to develop new drugs. And we've done that very successfully. We want to understand how these cancers develop. We don't really understand what causes the mutation in the first place, but what we know is once this happens, it sets the seed for more aggressive cancers to happen, and we're trying to study this. We want to study cell fate determination. Once a cancer develops, how does it evolve to become resistant to a drug? And I'll tell you a little bit about that in the next part of my talk. With Sarah, we work very closely using various genomic technologies such as ctDNA to understand how these cancers evolve. And one of the other things we've become very interested in is using epigenetics to make our immune system much more effective. To survey and eradicate these cancers. We've changed drugs so that they can now be used much like antibodies in the laboratory, so we can understand them in far greater detail. We can look into a cell to understand how much drug went in there, whereabouts in the genome the drug is going, and what tissues the drug is being distributed in. So the last part of my talk, I just want to talk about one of the areas that the lab is intensely focused on. When you heard Sarah's talk, you heard about how new mutations develop as people get exposed to therapy. And this, by and large, is how we think about resistance to cancer therapies. So we have our leukemia, we give the patient chemotherapy, they get a new mutation, and as a consequence, that new mutation renders the drug redundant, and then this cancer then comes back. And there's very, very good examples of this. So this is just a fish plot showing that this actually happens. But about 3 years ago now, we looked at how resistance develops to epigenetic drugs. And by and large, what we found is that when we look at the cancer itself, there are some cells that are very responsive, and these cancer stem cells, these red cells, are not very responsive. But when we expose them to the drug, we eradicate the blast, but the cancer stem cells survive, and they become the dominant population. Okay? In some ways, this reinforces the fact that we were actually able to show that resistance develops from cancer stem cells. But then in the last paragraph of this paper, I wrote this, because one of the biggest discoveries to us was the fact that this evolution from being sensitive to being resistant happened in the absence of any new mutations. There was no evidence that a new mutation happened. What it suggested instead was that the cancer cell said, we can't use this pathway, we'll use a different pathway. And that to me actually was pretty scary, because it meant that the way we were thinking about how resistance developed had to be rethought. Okay? What does that mean? So here are all our cancer stem cells, and the one in red is the one that survives. It could be that this was pre-existing, and it just happened to have the right gene expression program. When the selection pressure was applied, we got rid of everything else, and that survived. Right? The other option is that it wasn't pre-existing, But one of these cancer stem cells had a greater ability to adapt. So when we applied the pressure, this cell adapted quicker than its neighbors, and it was able to survive. Now you might think, why is this really important, right? Because understanding that process tells you how better to treat it, okay? And let me stop the talk by just giving you an example of what the lab is really thinking about. I'm going to start by explaining it using this streetscape, which is sort of semi-redundant now because we moved from where we were, okay? So this is where we used to be, Peter MacCallum Cancer Centre, and you can think about this, Flinders Street Station, as being your destination of being resistant to a drug. And we think about cancer much like you and I would behave, right? Which is that if you needed to get out, right? Pressure at work was really hard and you need to get out, you'll take the fastest route, right, to your escape. You'll take the biggest streets to get there as quick as possible. And you know, over the last few decades, we've worked out what these streets are and we've built drugs to them. And what we've learnt from many of these drugs is that cancers will mutate and make the drug redundant and still use the same streets, okay? So it'll just develop a mutation and say, I don't care about this anymore, I'm still going to go through it. It'll break through the barrier. Right? But now we've built epigenetic drugs, and they can't mutate. Okay? If they mutate, then they're actually getting rid of the way they work in the first place. That's how epigenetic drugs work. So now they do what you and I would do, which is say, okay, that street's blocked. Let's take the next main street and still get there quickly. And the temptation, now that we've started to work some of these out, is to go and say, okay, we now know what that is, and we'll just build drugs that block that. Right? But if you did that, well, the cancer cell will do what you and I would do, which is get really irritated because our two main streets to get out have been blocked, and it will be slow, but it will adapt, right? And it will take the side streets. Right? And you can keep putting up blockades, and I don't think that's very effective. Right? So what I think is the way we use epigenetic drugs is to make the cell do what is predictable. Work out what the second avenue is, right? And ask it to take the second main street and set a trap later on. Far less side streets, far less avenues to escape. You've got to get it before it gets to Flinders Street. And you've got to follow it along the way to know how far it's travelled, but let it make a predictable decision. So that's kind of a main concept that we've been thinking about in the lab. I'm going to stop there with the same slide that Sarah had. You know, in part we run two very, very different programs of research that are highly, highly complementary. We can't, you know, I could not do the research I do without her skill set and without the experience and expertise that her lab members bring. And so even though we run two separate labs, we work as one big team. And that, by and large, has been the reason we have had a modicum of success over the years. So I'm going to stop there.
Don't go away. I just wanted to thank Sara-Jane and Mark for just a wonderful example of how they communicated a complex subject in just such a wonderful way that we could comprehend and obviously stimulate some questions. So thank you, Sara-Jane and Mark. And finally, to thank our partners and supporters. Without them, these events wouldn't be possible. They've been with us, some have been with us for 10 years, but irrespective of that, they make these events possible. So please join with me in thanking them for their generous support. We look forward to seeing you at the next event. Thank you and good evening.